All right, now it's time to change gears and move a little bit away from transmission genetics. We're never going to get away from transmission genetics in a genetics course completely, but now we can actually start thinking about the next step down. Remember, we started from macroscopic to microscopic. That's how we're traveling in our analysis of genetics. And so we started with, a little bit of population genetics, then transmission genetics, and now we're going to move down to the microscopic level. Now, not the submicroscopic level, that's going to be molecular genetics, but the microscopic level, and that's going to be cytogenetics. So we're up to chromosome variation, and so this is the start of cytogenetics. All right, let's do a little bit of a refresher before we get into it. So first and foremost, chromosome number in the nucleus is specific to each species. It's a product of evolution. So for example, fruit flies have 8 chromosomes, corn plants have 20 chromosomes, humans have 46 total chromosomes, right? 23 pairs. And realistically, however many chromosomes you have does not indicate intelligence or, sophistication or more advanced species or a less advanced species. It really is just a product of evolution. So since we're talking about humans having 46 chromosomes, let's refresh, right? So we are diploid. Diploid means we have 2 sets of each chromosome, or 2 copies, I should say, of each chromosome, same thing. And so we have 2 of each autosome. Those are the numbered chromosomes. And then we also have 1 pair of sex chromosomes. So we have 2 sex chromosomes, right? And so wild type would be XX female, XY male. And so that makes it 23 pairs total, 22 autosome pairs and 1 pair of sex chromosomes. Now, the reason why I'm going over these numbers again, because of course we've done them many times over, and so you probably are very comfortable with those numbers. But from here on out, when we start talking about chromosome number and changing the number of chromosomes, adding a chromosome, taking away a chromosome, that's going to be very important that you remember the numbers, because you're going to see new numbers. And those new numbers you have to be able to identify right away as saying, oh, well, 47, that's 1 extra chromosome. And so that we will see is going to be something called an aneuploidy. Or if there were 69 chromosomes, well, that'd be a whole extra set of chromosomes, 23 times 3. And so that would be an example of polyploidy. Or if it was, 45, we're missing a chromosome. That's also another example of an aneuploidy. So we'll get to what those things are in just a moment. Let's go over a couple last things about chromosomes so that you're all on the same page. We're all on the same page. So remember again, our somatic cells, our body cells are diploid. They contain 2 copies of every chromosome. Something that a lot of students get confused is our somatic cells contain the sex chromosomes too. So they have 23 pairs of chromosomes total. They've got the 22 autosome pairs and then the 1 pair of sex chromosomes. And so our sex cells, right, those are the ones that are producing gametes. They're the ones that, well, there you go, they produce gametes. They produce haploid gametes with only 23 chromosomes each. And we saw that during meiosis. So this is the tissue that you find in testes and ovaries of human beings. All right, just again, a little bit of a refresher. Remember that chromosomes condense tremendously, such that they can then become clearly visible during early stages of cell division, right? So during prophase, they start to condense down. And so most of the life, like all of interphase, the cell's nucleus looks like this. You can't really make out the chromosomes. We stained it so that you could actually make out the 2 copies of chromosome 4 among all the other chromosomes just mixed together. So they're not really discernible most of the life of the cell, but then during, either M phase of mitosis or meiosis, they condense down into readily identifiable structures. So in cytogenetics, that's the time at which we are going to take a look at them. We're basically going to look at them after they've been replicated in early prophase or arrested at metaphase, but we are not going to really pay attention to the fact that they're replicated. We don't care about that. We're not looking to them mitotically dividing or meiotically dividing for that matter. It's just the best time to take a look at the chromosomes, to keep track of the chromosomes, to just analyze the chromosomes in general. And so we are going to see a lot of karyotypes, and those karyotypes are taken usually with the chromosomes arrested at metaphase because of the addition of a poison that causes the cells to basically just kind of freeze once they get to metaphase. Nothing else happens. So they would die off, but at that point we fix them and then we put them on a microscope slide and look at them. So human chromosomes, oh, here's a good example of that, right? So this would be a cell that has been treated with a number of chemicals so that we can visualize certain things. Like you could see that we've got the spooky little centromeres, right? And then actually, there was one day that I gave this lecture on Halloween and it was perfect. But obviously that's not today. So what we've got is you can see the centromeric DNA. You can see even there's some centromeric DNA before this nucleus, the DNA has fully condensed down. Here it's all condensed, and so we can start to see all those chromosomes. And so human chromosomes are usually studied while they are in metaphase of mitosis, because again, we have a poison that we use that was isolated from obviously a poisonous organism, and it arrests cells in metaphase, and so then we use it. It's called colchicine. So they are condensed into identifiable structures, right? Those X shapes. So they've already gone through S phase of interphase, and so they're in a replicated state. So we're not really going to pay much attention to that, right? We're just going to treat this as one chromosome, even though it's one chromosome in its duplicated state. We don't really have to worry about the ×2 and the duplicated state and all that stuff because again, this cell isn't going anywhere. This is it. We take a picture, and at that point, it's essentially it's fixed on a microscope. It's dead. So this is just to kind of analyze what's going on in these cells. So they're condensed into identifiable structures, that characteristic X shape. And remember that these are in the replicated state. But again, that is just, I can't stress this enough. It was so important when we were talking about mitosis and meiosis. Now, it is completely unimportant. It is just an artifact of the process in which we photograph these chromosomes. We photograph them in their most condensed state. That's after they've been replicated so that they condense down in mitosis, right, or meiosis, so that they condense, generally it's mitosis, right? We're dealing mostly with somatic cells. But they condense down into their characteristic, two sister chromatids attached at the hip, right, at the centromere. And so they are going to look like that. So we don't really care. I can't say it enough. We don't care that they're in their replicated state. This is just one chromosome that we're going to be looking at. Here's another chromosome we're going to be looking at. We're going to ignore the replicated state. And ignoring the replicated state, the earliest way to analyze these chromosomes was using GEMSA stains. So remember way back, we're talking about the 1900s, early 1900s. So Walter Fleming was the one who identified thread-like bodies that turned out to be chromosomes. And then later, the reason why I always have to take a pause and think about that is because then later Walter Sutton was the one who linked them to Mendel's work. But Walter Fleming identified these chromosomes. And way back then, we didn't have all the sophisticated techniques we have now. And so there was a stain that was used to basically stain these chromosomes. And so that's GEMSA stain. And so GEMSA stain stains the chromosomes either dark blue or light blue, depending on, actually, believe it or not, depending on what sequence is present. So, and that is going to, and when I say what sequence, it really is how well the stain can penetrate into that chromosome. And so remember, G's and C's are going to, well, I don't know if I mentioned this before, but G's and C's hydrogen bond 3 times each. There are 3 hydrogen bonds between a GC base pair, and then there are only 2 hydrogen bonds between an AT base pair. So you can think of ATs as a little looser. And so GEMSA stain can get in there better. So GEMSA stain can't get into GC-rich regions as well, and so that's where you're going to have light blue as opposed to the darker blues. And that becomes relevant, I think I have it in the slides coming up, but that becomes relevant because in the human genome, believe it or not, our genes are more GC-rich than AT-rich. So that gives you an insight as to where you're going to find a bunch of genes. And I think I told you, right? So I used to work at Lawrence Livermore National Laboratory. When we got chromosome 19 to sequence in the Human Genome Project, everybody was bummed because everybody wanted chromosome 1. It was the biggest. It was going to be theoretically the best. And so then it turned out to be okay because chromosome 19, while it's a little one, it is the most gene dense. And along the lines of our discussion, notice how much light blue it has. There's a lot of genes packed onto chromosome 19. And you can see that from the G banding, from the GEMSA staining. We call this G banding as well. Now, all of that I just told you is stuff we learned later. So let's go back to the beginning. So GEMSA-stained chromosomes show unique patterns. So chromosomes were identified based off of their GEMSA staining patterns. So if you were looking at these a lot, you would be able to identify chromosomes by their patterns. And also, of course, their size, right? So you could see that this chromosome is clearly different than this chromosome. So they're 2 different chromosomes. And so these 2 chromosomes are similar in size, but the banding patterns are different. So those are different chromosomes. And then, of course, they're also identified by the location of their centromere, right? So, like, for example, well, I could make an argument that this chromosome 3 and chromosome 4 are pretty close in size, but the centromere is very different. Here we have a metacentric chromosome. Here we have a submetacentric chromosome. So, gem sustaining yields G-banding. G-banding identifies regions of low gene density because, as I said a moment ago, it stains very well areas rich in A's and T's because A's and T's only have 2 hydrogen bonds between them. And so each arm of a chromosome is going to be further subdivided into regions from the centromere. Now, this is just a cartoonish image, but if you ever look up a gene, say, for example, you go to OMIM, which is Online Mendelian Inheritance in Man, that's a definitive source for genetic information. It's curated by Johns Hopkins, the same Johns Hopkins. Actually, I take it back. It might be curated by Tufts. I have to look that up. But it's a curated resource. It's way better. Let me just say it this way. It's way better than Wikipedia. And so if you ever look up a gene there, you get all sorts of, all sorts of information from literature to all sorts of different things. Peer-reviewed papers is what I mean when I say literature. And then, of course, you get the location of the gene. And so you get we've sequenced the human genome, so you can get the exact location of the gene. But you can also get its chromosomal address. And that's what this looks like. When you run into something like this is a very simplified version, but this is, okay, chromosome 1. It's on the Q arm, right? It's not on the P arm. It's on the, so remember, P is for petite, Q is for the larger arm. P is usually the P arm, the smaller arm is on top, larger arm is on the bottom. And so it's on the Q arm, and then it's in, basically you could see like region 2, band 4, or something like that. So you have to, obviously the picture here doesn't really do it justice because it's just a random picture that somebody threw together. But basically, each arm is subdivided into regions from centromere to the end. So from the centromere to the end, see how we're counting 1, 2, 3, 1, 2, 3, going that way. And so I said, as I said a moment ago, 1Q 2.4 is an address, if you will, for a locus. It's a gene locus. And so it's a locus designation, if you don't want to use the word address. And so it's telling us, like I said, chromosome 1, long arm, and then region 2, band 4. And so obviously real, loci are going to have usually quite a bit more information after the arm, right? So it'll have like lots of numbers, dot, 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 dot. So each, you just move down to a more specific, like this, region 2 could be, region 4 could be subdivided into multiple different bands if you look at it more closely. And so therefore it could be 4 point something, that sort of thing. All right, so here's an old-fashioned karyotype. So, obviously things were taken in black and white, but that's gem-sustaining. So that's G-banding. These chromosomes they existed. You've seen this one before, I think. But these chromosomes were in a cell, and so they basically take a picture, and then you just rearrange the chromosomes, line them up. Obviously they're not going to just line up like little strips of bacon. But basically you line them up, and now you have your chromosomes and in a karyotype. So this is called a karyotype. So for convenience, images of chromosomes are arranged into pairs according to size, centromere location, and G-banding. And that forms a karyotype. And you can learn a lot from a karyotype. So like this karyotype tells us, okay, well, we're dealing with an XX female. And if we were to examine it much more closely, we could see whether we're missing any sections on any of these chromosomes or, some of them, the staining didn't work as well. So like here, the staining, you got a little too long with the exposure to the stain. Imagine like this one, this one, this one, they're all darker than the rest, right? So imagine the stain penetrated from like one direction, and so it then it diffused, across the nucleus. And so these were closest to the point of entry of the stain. And so they were exposed to, the stain was washing over them longer than it did to get to like, stain 19 or 16 or the lighter colored ones. But again, remember we talked about 19 being lighter because it doesn't absorb stain as well. So, so little artifacts like this aside, the difference in the darkness of the stain, here they look pretty close in terms of their intensity. And we know, again, chromosome 19, very gene dense, and so therefore it doesn't pick up stain very well. It's got a lot of GC pairs. So karyotypic analysis of banded chromosomes is actually very powerful. I don't know why your book has omitted it. Remember from way back from, I think it might have been chapter 1 when we first started the course, I said that previous versions of this very text included the 4 areas of modern genetics as population genetics, transmission genetics, cytogenetics, and then molecular genetics. Now your current edition of this textbook only includes, population, transmission, and molecular. And so I don't know if they want to, say that, well, you're using a microscope, so this is molecular, but these are pretty large, these are macroscopic in terms of molecules. You could see chromosomes with the naked eye in certain cells, right? So like there are these things called Pachytene chromosomes in the, where is it? In certain, they're larvae, maggots. I think they're botfly larvae, so maggots. And so they have such large chromosomes you can actually see them with the naked eye. You don't even need, magnification. So, putting them into molecular is a little bit of a stretch. So that's why we're focusing on cytogenetics for the moment, because it's still very powerful. Let me show you. So karyotypes are constructed from all sorts of different types of cell samples, but they do still take time. And so if you ever wonder why, like if you have to get some sort of test done in the hospital, why it takes days to get your results, you would think, oh, everything's getting faster now and automated would be a lot faster. Well, turns out the way to make a karyotype is to take a blood sample or to take a sample of tissue, and then you have to grow it up in the lab to the point where you have a lot of those cells, and they're rapidly growing. You need them growing, and so that they are constantly turning over so that you have a plenty of them, that when you hit them with colchicine, with that chemical, with that poison, that they stop in metaphase, right? If they're not rapidly growing, then they're never going to make it. Like, maybe they're just sitting there in, G1, just hanging out, maybe G2, maybe S phase, whatever. They're not really doing much. So you need them rapidly growing. So it takes a little while to get them stimulated, and our cells don't grow the fastest in the world, right? They're not nearly as fast as bacteria. And so then once they're growing, you hit them with colchicine, and then you can arrest them at metaphase. And then you could, the colchicine arrests them at metaphase, and then you can stain, and then you can take a look at the karyotypes. So chromosome mutations are identifiable from these karyotypes. And so chromosome mutations can be grouped into three basic categories. So we're going to talk about chromosome rearrangements. Chromosome rearrangements are when the structure of a chromosome is altered in some way, but the overall number of chromosomes doesn't change. Then we'll talk, I mentioned this earlier, right? Aneuploidies. There it is in a bullet point. We'll get into it in more detail in a moment. But aneuploidies are when the number of chromosomes is altered such that you're just losing one or picking up one. So you're just adding or subtracting one or two or three chromosomes, not an entire set of chromosomes. If you had an entire set that you add or lose, that's when you talk about not aneuploidy, but polyploidy. Now, if you lose an entire set of chromosomes, that's fatal, right? So we don't really talk about polyploidy in terms of what happens if you lose a set and you're down to one set. Any somatic cell with only one set of chromosomes is going to fall apart. It's basically going to die. So when you pick up extra sets, you have many sets of chromosomes. That's why it's called polyploidy. One or more complete sets of chromosomes are added, right? When you subtract a set, cell dies, so there's nothing to really study there. If you add an entire set, that does cause severe problems in animals. However, in plants, polyploidy is a wondrous thing. It provides all sorts of, it's just like the plants are like, okay, let's try it, right? Give me a whole extra set of chromosomes. Let me see what I can do. And so it's very interesting. So there's a whole area of, these three different types of chromosomal mutations. So you could have rearrangements within a chromosome, or one or two chromosomes, or just one chromosome. You could have picking up or losing a chromosome, or multiple aneuploidies, right? You could pick up two or something like that. Or you could have polyploidy, where one or more complete sets of chromosomes are added. So let's start with chromosome rearrangements. There are four basic types of these mutations, and so we're going to go through them, not in this order, but there's deletions, duplications, inversions, and translocations. And then there are multiple versions of these different types of things. So this is the kind of stuff that I'm going to test you on, right? Like, what type of chromosomal rearrangement is this? And what type of chromosomal rearrangement is this? And it's not just what type it is. It's also, I'm going to ask you, and we'll go over this when we go through them individually. I'm going to ask you specifically what is bad about this type of chromosomal rearrangement. Why is it something that we should care about? Why is it something we should pay attention to? All right, so here is just a couple examples of them. We're going to go through them individually. But we have a deletion where you lose a chunk, right? So that's pretty straightforward. Duplication, you add a chunk. All right, so that's pretty straightforward. Inversion, you might not be familiar with inversions. It's where like it kind of breaks off, flips around, and goes back in. So notice it's 4, 3, 2, 1 going this way, and now it's 4, 2, 3, 1. So that little section was inverted. You could also have translocations. I've mentioned these a few times, right? Where you have non-homologous recombination. So two chromosomes that are not homologous swap parts where they shouldn't have. That's called a translocation. And so, well, I said two. So if you just have a chunk of a chromosome ending up on another chromosome and it's not, you know, this one that lost a piece of itself does not get anything in return, that'd be a simple translocation. Whereas what I just mentioned was a reciprocal translocation where both of them basically swap parts. They just exchange parts. That would be a reciprocal translocation. So let me go over these in a little bit more detail. So let's start with rearrangements. So well, there's all rearrangements, but rearrangements in chromosomal structure can involve one, two, or more chromosomes. And all of those that I just showed you, the way they occur is they result from breakage and reunion of chromosomal parts. So remember, our chromosomes can break. They can break from shear force. There could be certain areas that are fragile and that could actually break very easily. And then, of course, we have homologous recombination. And so sometimes you can have homologous recombination go awry. So breaks occur spontaneously through errors in replication, errors in recombination, all sorts of different things. And so, I mentioned, you know, you could have breaks due to environmental agents. So I mentioned shear force, but there's many more things, right? Radiation can cause double-stranded breaks of chromosomes. You might remember way back when I talked about Deinococcus radiodurans being discovered because it's an organism that could survive tremendous amounts of radiation when we were talking about Craig Venter and synthetic genomics. And basically, Deinococcus radiodurans has a repair mechanism where it can handle double-stranded breaks. Radiation is ionizing radiation, and it causes double-stranded breaks. So radiation, so that would be like gamma radiation, ionizing radiation, even UV light. UV light is it's not as harsh as gamma radiation, but it causes thymine dimers, and it can cause breaks because of those thymine dimers and just actual breaks if it's in strong enough intensity. Viruses, of course, they want to, you know, basically shove their genetic material into your genome. So the only way they can get their stuff into your genome is by breaking a chromosome and shoving their stuff in there, right? So that would be a virus, particular type of virus, right? A DNA virus. Then we have chemicals, right? So certain chemicals can cause double-stranded breaks. They can cause all sorts of breaks in our chromosomes. They can weaken our chromosomes. And so we'll talk about all these. If there's time permitting, we can get to the mutations portion of the course, and we'll talk about all the different ways in which our DNA is susceptible to mutations. It's very depressing, actually, how many mutations we pick up on a daily basis all the time. But the good news is we have repair mechanisms. So keep that in mind, that we have repair mechanisms that we can, you know, undo some of these mutations. Can't undo all of them. That's the process of aging. So, you know, I hate to break it to you. You're not going to live forever. I'm sorry. But you know, we do sustain ourselves for quite a while because we can repair a lot of these DNA forms of DNA damage. Okay, so let's start with a translocation. And I mentioned simple and reciprocal translocations. Well, here's a simple translocation. There's not much to it. You get a break here, and then that chunk of a chromosome ends up over here. And so you have a simple translocation. So imagine if you go back to, say, for example, when we were talking about let's see. We were talking about variations in Mendelian ratios, and we were talking about alterations of determining sex, right? So imagine you have a little tiny Y chromosome, and then it picks up a huge chunk of the X chromosome, right? And so in the process, let's say in that process, when we broke this chromosome, we destroyed some of the genes on the Y chromosome that might have been necessary. So technically, you have in an individual, you can imagine an individual has, you know, part of an X chromosome, the other part of an X chromosome. So this one individual basically has a fully functional X chromosome, except it's just in different segments, and then lost the function of their Y chromosome. And so what could happen there is you have an XY individual that essentially presents as an XX female. because of the translocation event here. Alternatively, this could be, silent, right? You could have, well, okay, nothing happened to the Y chromosome. It just picked up some extra stuff on the X chromosome. We still have a functional X chromosome between the two halves here, and we have a functional Y chromosome. So this is just an XY individual. So this could be me, right? So I've never had my karyotype done, but as far as I know, I'm a pretty normal male, and so perhaps I'm carrying a simple translocation in which there's no effect. Now, reciprocal translocations, on the other hand, usually cause some. Now, again, they could be silent. You could, somehow not disrupt anything. But more likely, what happens is that you create some problems. So let's talk about why these things are bad. What you want to think of are two things, right? There's two things that you want to remember. And so, and forgive me, I'm using a little mouse, not like a stylus, but basically, if you have, there's two things we're going to be looking for. And so we're going to be looking for gene dosage. This is probably not a good idea. And so gene dosage is one of the two things you want to really pay attention to, and I'll go over what it is in just a second. And then the other thing that you really want to pay attention to and you want to look for are the possibility of creating, I don't know, can you read that? So fusion genes. So yeah, so next time I'll just edit my slides and make sure it appears in a bullet point. You live and learn. All right, so anyway, so we've got gene dosage and fusion genes. I don't know, it might wake you up. So those are two things you want to look for, right? So now let's kind of think about these. So in the event of a simple translocation, did we pick up any extra genes? Like imagine if we duplicated a chromosome, we had a big extra chunk of a chromosome and the genes in that section were duplicated. Well, then we would pick up extra gene dosage, right? Think of like medication, like you have a certain amount of medication, you don't want to double your dosage without knowing what's going to happen, right? So generally you just don't want to double your dosage. So we don't have any gene dosage issues here. We have maybe a very, very slight gene dosage issue because, when you make breaks, you could disrupt a particular gene. So we don't really care about gene dosage when it comes to translocations. But with translocations, what we do care about is the formation of fusion genes. Fusion genes are going to be pretty bad. So fusion genes are when, imagine that you had like a gene that, let's say a gene started here, and was going, and then we swap in a different part. Oh, I guess I should be using this one. So it started here and then it was going, but now it is down here, right? So it's way over here. I guess I should have taken this section. So it started here and it's going this way, but now it is here and it goes into the blue. Right? And so basically what you're creating is, you imagine if you had the start of one gene and then after the translocation event, it's connected to the end of a different gene. That can create problems. That creates a fusion gene, and these fusion genes can be catastrophic. So here, let me give you an example of a hypothetical. Imagine you have a gene responsible for a lipase, something that destroys lipids or phospholipids. And this gene is supposed to go into a vacuole or a lysosome, basically into a digestive body within the cell. So you're making your enzyme, your lipase. And the second half of it causes it to translocate to the lysosome, right? It directs it to where it needs to go into the cell. But then you create a fusion gene with a kinase that basically phosphorylates things on the inner leaflet of the plasma membrane. So if you take the enzymatic activity of a lipase and then the localization part of the gene, the second half of the gene, that basically targets it to the inner leaflet of the plasma membrane, so the inside, that is the cytoplasmic or cytosolic side of the plasma membrane, the intracellular side, and you make a fusion gene out of that, well, then that means that you're going to now target a lipase to the inner leaflet of the plasma membrane. And so what's it's going to do when it gets there but just chomp up your membrane, decrease the integrity of your membrane and basically cause the cell to die. So you could imagine these fusion genes, there's all sorts of different scenarios where they're pretty bad. And I'm not just making this up. Here's some examples. So AML1-ETO is a famous fusion gene. There's two of them, in fact, that are responsible for leukemia. So the AML1 protein and the ETO protein get fused in a fusion gene when you have an 821 reciprocal translocation. Wow, that sounded very genetic, and you understood all of that, right? Chromosome 8, chromosome 21, reciprocal translocation between the two. When that happens, you create an AML1-ETO fusion protein. And believe it or not, it is present in 12%. Now, that may not sound like a very high number, but that is a massive number. We're talking of all the, acute myelogenous leukemia cases that spring up out of nowhere de novo, right? So these are individuals that have no history of it in their family or anything like that. They're not exposed to tremendous, carcinogens or anything like that. They just all of a sudden they have acute myelogenous leukemia. Where did that come from? 12% of those cases have the AML1-ETO fusion protein. So there's a correlation between the AML1-ETO fusion protein and 821 reciprocal translocation and acute myelogenous leukemia. There's another one. I don't know if I have it on the next slide, so or in the next bullet point, but there's another one between 9 and 22. And so that one is for CML. So that's chronic myelogenous leukemia. All right, so now let's switch gears to duplications. So you tell me, is this an issue of gene dosage or fusion genes? So here are what we have. We have a transition from 4 to 3, so 4, 3, 2, 1. Now we have 4, 3, 2, 3, 2, 1. So really, okay, so if we're talking about fusion gene formation, 4, 3, 2, 1, 4, 3, 2 is nothing new, 3, 2, 1 is nothing new, but the 2, 3 transition, okay, that's new. So maybe there's this slight possibility of a fusion gene being made at this new junction. However, what we definitely have a problem with here is going to be gene dosage. Because imagine that you have, let's say you have 2 of these chromosomes, right? And so this is a duplication just happening in one. The other, the homologous chromosome, let's say this is the paternal chromosome that you have. The maternal chromosome is just fine doing its own thing. And so the paternal chromosome undergoes a duplication event. And so if that's the case, now you have the equivalent of 3 of the genes that are in sections, the section between 3 and 2, right? So look, 3 and 2, 3 and 2. So you have 2 of them on here, and then you have 1 of them on the other homologous chromosome. So imagine that these genes are housekeeping genes that are expressed at a very high level. So that's called, if they're turned on all the time, being expressed all the time at the highest level, that's called constitutive expression. And so imagine that they're constitutively expressed. If they're constitutively expressed, you just increased that gene expression by 50%, right? You had 2 of them, each making up, they're both being expressed at 100%, and then you just added another gene to the mix. So now you're, I don't know. Before you were having the equivalent of 100% off one chromosome, 100% off the other chromosome, so that grand total 200, right? But now you're getting 200% off of one chromosome, 100% off the other chromosome, and so that could be fatal. That could be too much gene dosage. Remember, too much gene dosage is a bad thing. Too little gene dosage is a bad thing. Regulating our genes, we talk about this in an entire chapter, the regulation of gene expression. It's a very, very, very complex process, highly complex process that needs to be regulated on many levels. And so it's very important that we maintain the proper gene dosage, and duplications are bad because they increase gene dosage. So here's how I would kind of depict it on like a test question, right? So you have, this is using images. We could certainly use images, or you could just keep it simple, right? If you're just typing it out, so the little dot represents a centromere, A, B, C, D, E, F, then E, F again, right? G, H, I. So if I didn't underline it and you say, hey, what happened there? You'd say, oh, that looks like a duplication. Now, this is what's called a tandem duplication because there's like 2 EF segments in a row. You could have a displaced duplication where it's EF, G, there's the other EF, right? So it actually got duplicated and then placed somewhere else. So now if you have a displaced duplication, so tandem duplication, minor fusion gene worry, right? But mainly gene dosage. Displaced duplication, however, is going to start to increase your concern about fusion genes because we now have a GE boundary that we never had before and an FH boundary that we never had before. And then we still have our regular gene dosage issue as well. Here you can see here is a reverse duplication. So again, we're increasing gene dosage certainly, and we're starting to raise our antenna on whether we have fusion genes or not. And so basically the question is, how on earth do all of these types of duplications occur? And so it's actually, if you study recombination, if you study DNA synthesis, it's actually pretty easy to see how this could happen. So let's talk about misaligned chromosomes. So basically, remember, we need our chromosomes to synapse during meiosis, right? So meiosis 1. And so this is why we had to talk about, meiosis and mitosis in such detail early on in the class. They just never go away. So if we're talking about prophase 1, the homologous chromosomes are condensed and they are now replicated and they're going to line up in synapse. Well, what if they don't synapse perfectly? What if they're misaligned? And then a crossover event occurs. And so you can see that we have non-equitable recombination. And so it's still technically a form of homologous recombination because we have homologous chromosomes, but if they're misaligned, then you end up with a duplication in one chromosome. And actually, it tells us how we get our deletion in the other chromosome because it was an inequitable exchange. And so it all depends on, so this is meiosis, right? So it all depends on whether this is going to be used in, fertilization, whether this is going to be used in fertilization, whether this is going to be used in fertilization, whichever one of these end up in the gamete that is going to, either be fertilized or fertilized. Those are going to determine whether nothing, this could happen, and then, who cares if this ends up being the chromosome we use out of these 4. Or, if this ends up being the chromosome we use out of these 4, it doesn't matter. But if we use this, then we have a duplication. If we use this chromosome, right, just by chance, that's the chromosome that ends up in the gamete that ends up being involved in fertilization, then that is how a deletion occurs. So we actually described how we come up with duplications and deletions in trying to figure out how duplications occur with misaligned chromosomes. All right, let's talk about inversions. So here's our normal chromosome, A, B, C, D, E, F, G, H, I. And there are different types of inversions. Inversions that do not involve the centromere are called pair-acentric inversions. And so you can see that here, right? So A, B, C, D, E, but it looks like B, C, D, E are going to basically break and then swing around and come back in. And so that's a paracentric inversion. Now, of course, if it does involve the centromere, the easy way I remember trying to remember these when I was first learning them is paracentric, meaning around the centromere, right? So not involving the centromere. Or you could even say away from the centromere. And then pericentric inversions. Pericentric inversions are going to involve the centromere. And so you can see that here. So the inverted region is basically surrounding the centromere. Now the last one we'll talk about is, one thing I forgot to mention is that inversions, you don't have any gene dosage issue, right? It's minuscule, but of course those inversions do certainly raise our concern about fusion genes. Now let's actually catch up, right? So now we're at deletions, and so deletions are, in my opinion, the worst possible chromosomal mutation you can get. And I say this because, and it's arguable, people can argue different things, but here's my take on it. Millions of years of evolution went into creating and constructing and crafting the sequence in this portion of the chromosome, and then it gets cut out and destroyed and it's gone forever. That sounds pretty bad to me. So I would argue deletions are by far the worst type of chromosomal, rearrangement mutation. And so we certainly have a gene dosage issue. We lack gene dosage. We lost all the genes in segment C. Remember, this is a chromosome. This is not just like one gene in this area. There could be multiple genes in this area. And so you're losing that. You're also, you also introduce the possibility of a fusion gene because now B and D have access to each other. But realistically, who cares about the fusion gene? You just deleted a section of a gene, a section of a chromosome. So you lost multiple genes. And so, like I said, so there's different types of deletions. So you could have like a single break, and then you lose, like you could say a single break. So we lose. This would be a terminal deletion. We lost a little chunk of the end. You could have two breaks and then reattachment of the outer pieces, and so that would be what's called an interstitial deletion. But really, so what I showed you on the previous image was an interstitial deletion as opposed to a terminal deletion. But realistically, both of these are bad, right? Chromosomal deletions are arguably the worst scenario because genetic material is lost forever. It's gone forever. I mean, even in an inversion, however unlikely it is that it's going to break in the same two places, flip back around and go back in, at least that's still a possibility. Whereas with, a deletion, that genetic material, once it's cut out, it's degraded. Key word here is degraded. And I can't stress that enough. Degraded means, gone forever. So that's gone, it's lost and gone forever. Millions of years of evolution down the drain. So basically, I would say deletions are the worst. And so here are some examples, right? So now if you're looking at a karyotype and you know what you're looking at chromosome 5, and you line up their centromeres as best you can. You line up the bending patterns. It's really hard to see in this image, but see how it's darker in this section, and then you line up their centromeres. Those are really closely lined up, and there is a significant chunk of the chromosome missing here. So this is actually, what is that? That's a terminal deletion. Terminal deletion of chromosome 5 can yield cri de chat. Cri de chat, if you speak French, then you know that this means cry of the cat, and that's because when the baby is born, it makes a very shrill, like a cat in pain sound. Does not, sound like a normal baby, and that's because you need this section of chromosome 5 for proper larynx development, vocal cord development, and also for cognition, and so you're going to have a child with cri de chat. That child is not going to be able to basically be fully functional, not going to be able to speak at all or properly, just making noises because the vocal cords have not been they're not there, right? They're not formed properly, and so they cannot be utilized for language. So that's an example of a terminal deletion.